Hello students, welcome to another lecture of human anatomy and physiology, and in today's lecture we will be studying about haemopoiesis or hematopoiesis. So let's start. So as the name suggests, haemopoiesis is basically the process of formation of the cellular components of blood, right? It is the process by which the immature cells, they develop into mature blood cells, right? Monophyletic theory is the currently accepted theory on how exactly this process works, and this theory simply says that a single type of stem cell is responsible for giving rise to different blood cells, right? And the stem cell which is responsible for giving rise to different blood cells is termed as pluripotent stem cell. Now haemopoiesis, as I told you earlier, is the process of the formation of all the cellular components of blood, and it occurs in the bone marrow, spleen and liver. Then the body continuously manufactures new blood cell to replace the old one, right? And the rate of haemopoiesis, it depends upon the body's need, right? So as I told you in my last lecture also, that RBCs, they live for a period of 120 days, but the WBC, they survive only for a few hours. So depending upon the need of the body, the haemopoiesis or the rate of or the frequency of haemopoiesis varies. Now the process of haemopoiesis begins with unspecialized stem cell. These unspecialized stem cells, they multiply and they develop into precursor cell. Precursor cell, they further give rise to a specialized form of blood cell. Now this is the image showing the life cycle of your erythrocyte or the life cycle of your RBC. Right? So there are erythroblasts in the red bone marrow. With the help of dietary folic acid, vitamin B12 and dietary iron, hemoglobin is formed, with the help of which reticulocytes are formed and they circulate in the blood for 7 days and they mature there. Right? So after 7 days, erythrocytes are matured, right, and they live for a period of 120 days. Reticulocytes are basically immature RBCs, right? So after 120 days, RBCs are destroyed by a process termed as hemolysis in the spleen, and the iron content is recycled and the bilirubin is also produced after their destruction. So the bilirubin, which is a waste product for the body, is secreted in the bile and is further excreted from the body. Now under hematopoiesis, there are three processes, from which the first one is erythropoiesis. Erythropoiesis is the formation of RBC, and this process is triggered by a hormone which is termed as erythropoietin, right? So erythropoiesis takes place in the bone marrow, where hemopoietic stem cell, it forms proerythroblast. Proerythroblast differentiates and further forms reticulocytes. Reticulocytes are the immature RBC. They circulate in the blood and they mature to form RBCs. Second process under hematopoiesis is leukopoiesis. So leukopoiesis is the formation of white blood cells or leukocytes, and the hormone which is responsible for their stimulation is colony stimulating factor. Right? So the development of each kind of WBC begins with the division of hemopoietic stem cells into blast cells. Right? So there are three subdivisions of blast cells, one being your myoblast, second being your monoblast, and the third being your lymphoblast. Myoblasts, they further divide to form neutrophil, eosinophil and basophil. Monoblasts, they further develop to form monocytes, and lymphoblasts further develop to form lymphocytes. Last process under haemopoiesis is your thrombopoiesis. Thrombopoiesis is the process of formation of platelets, right? And this process mainly begins by formation of megakaryoblast. Megakaryoblast, they further divide to form megakaryocytes. And as I told you in my last lecture also, that platelets are not exactly cells, but they are fragments, right? So then megakaryocytes, they further develop into fragments which further, form your platelets. So this is an image showing the development of different cellular components of blood from your pluripotent stem cells, right? So thank you so much, dear students. This was all in today's lecture. If you have any queries, any doubt in today's lecture, please feel free to reach out to me. Thank you.